Hey guys, welcome to Biology Lesson 5.06 on meiosis. So what we're going to do in this video is we're going to take a look at the difference between mitosis and meiosis from a division standpoint. And we're going to be using some highlighters. I think those show up better. So if you want to grab highlighters, you can. So first we'll go back through mitosis and then we'll go through meiosis. But remember, meiosis is the process of having, that's cutting it in half, genetic information. So what we have here is we have an organism that has one homologous pair. So let's just say we have one homologous pair. And I'll usually do a dark blue and a light blue, but in this case, I'm going to actually use different colors to help us keep track of our chromosomes. So I'm going to do one of the chromosomes in blue highlighter. I'm going to do one of the chromosomes in pink highlighter, but this is a homologous pair. Okay, and at each point, I'm going to be asking you how many chromosomes, how many chromatids. So let's start off with mitosis. Here we go. Got my blue one and I've got my pink one. Could make that pink line nice and dark. So this is going to be interphase because I actually wouldn't see the chromosomes, but here we go. Now I'm duplicated. So how many chromosomes? This would be the beginning of prophase. So this is going to be prophase. How many chromosomes do I have and how many chromatids do I have? Okay, did you say 2 chromosomes? If you did, that would be correct. But I've got 4 chromatids because they're copied. Now I'm lined up in the middle. So what phase is that going to be? Good, metaphase. Here I have the blue pair being pulled apart, and I have the pink being pulled apart. And I end up with there we go. Remember, what am I? If I have 2 chromosomes that are homologous, what is my condition called? A lot of review questions in this one. So this cell starts out diploid. And look, I have an exact copy. So I have two exact copy daughter cells. And these are also diploid. Well, right away, you can tell with meiosis, I've got a lot more going on. I'm going to start off the same way. I've got blue, and I've got a pink. We're going to copy those. So I've got a copied blue and a copied pink. Now here, this is unique. So here we have got something special going on. This is called crossing over, and we're going to note this out. We're going to pull this out and talk about this. But right now, I just want to focus on the flow. So these guys get nice and cozy. In fact, they kind of bump up against each other. And we get weird things happening here. So we get they cross over, they crisscross. But for the flow, I want to pretend like that doesn't happen. So here we've got, now they separate. Instead of lining up in the middle, so we go through 2 divisions. The first division, and this is called meiosis 1. We use the Roman numeral 1. And for the metaphase, for meiosis 1, they line up side by side. So they line up side by side. And this would be the start of meiosis 2. So we can do a line, a dashed line. So in the first one, we've got basically one copy of a homologous pair goes one way and one copy goes the other way. So instead of lining up in the middle and pulling everything apart, we now have one homologous or one chromosome here and one chromosome here. So at what point does this become, at what point in this process, let's write this down, this is important enough. At what point does the cell go from diploid to haploid? All right, let's go back to the beginning. How many chromosomes here? 2 chromosomes. How many chromosomes here? 2 chromosomes, 4 chromatids. We're crossing over, we're beginning to separate, and now we come down here. How many chromosomes in that cell? Oh, 1 chromosome here, 2 chromatids. So it is at this spot, right here at the beginning of meiosis 2, that we go from diploid to haploid, or from haploid to diploid. Diploid to haploid, I said it right the first time. So the answer is the beginning of meiosis 2. And look at our end product. We have 4 cells that are produced that have half the genetic information. They have half. And we need that because if you try to combine these two, you would end up, so if you try to do fertilization here, you would end up with 4 chromosomes and that wouldn't work. So this would be mom set. We've got mom set here. This would actually be dad set. And you've got a set over here that belongs to mom. And you're going to combine these randomly. But from beginning to end, so what are our takeaways? We're starting off with a homologous pair. We did one in pink and one in blue. Even though they're homologous, I wanted to make them look very different. And we're only considering an organism that only has 1 homologous pair of chromosomes or 2 chromosomes. We've got chromosome 1, chromosome 2, it's a homologous pair. We copy the genetic information in prophase. We end up with 2 chromosomes still, but 4 chromatids. And at the end, we have exact copies. In meiosis, we start out the same way. We're doing the same thing. We get this weird crossing over business going on that we're going to pull that out and talk about that because that's super duper important. But in metaphase, instead of lining up on top of each other, they line up side by side. And when they separate, the homologs have separated. So this guy has the pink one, the blue one, this guy's got the pink one, and then it splits again, and we end up with a set that have half the genetic material. So at the beginning of meiosis 2, we go from diploid to haploid, diploid to haploid, diploid to haploid. Okay, that's an overview, and we're going to talk a little bit more about meiosis in the next video and crossing over. That's all for now.